Howdy, folks. Thanks for checking into Mr. Ulrich's Land of Biology. I am Mr. Ulrich, and we've talked already about how genes code for proteins, and we've even talked about how the environment can turn genes on and turn genes off. What we haven't really talked about are the mechanisms by which that can happen, and how that influences stuff like multicellular development. So in this notes cast, we're going to look into mechanisms of gene control, how to upregulate, downregulate, and some of the differences in how this works between eukaryotes and prokaryotes as well. We have a couple essential questions here that we need to be able to answer by the end of this. So how are genes turned on and turned off? And in order to really understand this, we really have to have a good handle on transcription and translation and the way DNA is stored. So if you still have questions on that, make sure that you go back and watch those videos. We know that all of the cells in animal come from the union of a single sperm and egg, and so they all have exactly the same genes, and yet through the process of development we get different cells turning on different genes and turning off different genes, and so there has to be a way that this can take place so that we can end up with complex babies like Bruce. To get an evolutionary perspective on gene regulation, it's helpful to look at how most primitive organisms, like prokaryotes, deal with turning genes on and off. It makes sense since bacteria, you can think of them almost as like are selected in that they need to be able to kind of adapt to new environments quickly. So that they can take advantage of resources that might be there and then might not be there. And so gene regulation becomes an adaptive advantage, so that they can kind of tailor their protein production, right? Which enzymes and other regulatory proteins are they making, so that they can match the environment that they have. So you only make the enzymes when needed, and you can turn them off when you don't need them. When we look at this in eukaryotes, especially multicellular ones, it's a little bit different. Rather than turning on and turning off genes in order to exploit resources in the environment, kind of matching the internal environment to the external environment, essentially, multicellular organisms kind of force the internal environment to stay the same in a variety of different external environments. So this is going to need some coordination between different parts of the body. So especially when we start talking about growing the parts that are going to be needed in order to do this regulation. So specialization is a long-term process involving whole groups of genes that are going to be turned on or turned off permanently in some cells as well as in other cells. And this is all really to allow the collective to survive. Some cells are actually, dispensable. If we think of epithelial cells, skin cells, their job is really to die to protect the cells that are behind them. When thinking about how genes are turned off or how genes are allowed to stay on, it's sometimes helpful, I think, to think about gene expression, right, going from DNA to a phenotype as like an assembly line. Information in a variety of different forms, different molecules is kind of passed along the line, and it's messed with by molecular machinery, cellular machinery, until you get, bing, a phenotype at the end. And if we think about each one of those steps of the assembly line, we can really think of a place where we can kind of flip the switch and turn it off. So we can we can affect the DNA. We can allow it to be accessible or not in the first place. And even if it's accessible, I can affect whether the proteins for transcription are there or not. And even if the information is transcribed, that RNA has to be processed in order to be able to be read by the ribosome. So I can affect the RNA processing, and even it has to get to the ribosome as well. And so I can affect the transport, how well that messenger RNA or how long it lasts until it gets to the ribosomes. And even if it gets there, the ribosomes have to be able to do their work. And so there's another spot spot that we can turn off the process. And even if the proteins are made, they have to be folded oftentimes into a particular shape. And so I can affect that process, turn that process off, and shut off the phenotype. And then I can even go ahead and kind of call over the garbage men. There are proteins and cellular machinery whose job it is to kind of hoover up and break down old proteins. And so I can label the proteins for garbage pickup, and then they get broken down, and then they're not there, and you don't get the phenotype. I have essentially turned off the gene. I want to take a couple moments to go through each one of these control points. The first one, making DNA accessible. We need to review how all of that DNA is crammed into the nucleus in the first place if we're going to understand how it's uncoiled to make it accessible, or not if I'm going to turn the gene expression off at this level. Right, so yeah, we get the double helix, that's a twist, and then that's coiled up into a coil, and then that coil twist is wrapped around nucleosomes, or it's wrapped around histones to form a nucleosome, and this string of nucleosomes makes up the chromatin fiber. I take the chromatin fiber and weave back and forth into these loop domains, take the loop domains and kind of weave them back and forth, and you end up with the chromosome. Taking a little closer look at the nucleosomes, those beads on the string themselves. It's made up of DNA wrapped around histone proteins. There are 8 histone protein molecules that make up each one of the nucleosomes. The proteins have positively charged side chains on the outside of them so that they can bind to and interact with the negatively charged DNA. The control of gene expression comes in by how tightly that DNA is wrapped around those histones. If I wrap it around really tightly and then it can't unzip and I can't get a transcription bubble, and I essentially turn off any of the genes that are associated with the DNA that's tightly wrapped around those histones. So that means in a specialized cell, I can have some genes that are still just wrapped right up in their histones, around their histones and their nucleosomes, and other pieces of DNA that are kind of unwrapped and ready to be transcribed. And we do see this in kind of the 2 different types of chromatin in the cell, be it heterochromatin or euchromatin. In order for DNA to be allowed to be unwound from the histones, the histone has to be acetylated so that transcription can actually take place, effectively turning the gene on. This happens by, just like it says, attaching an acetyl group to those histones, causing a conformational change and allowing those transcription factors to get at the genes themselves. Speaking of unwinding DNA and allowing transcription factors to get access, remember when we talked about transcription, we introduced this idea that there's more to a gene than just the section that codes for the protein. There are also all of these other sections that are involved in allowing that section, that coding section, to be coded for. We have the promoter region, which is relatively close. It's going to give a spot for the RNA polymerase and other transcription factors to attach and actually start the process. This is kind of like just the basic rate. As long as it's there, it's going to be happening okay. But there's other places further along, further away, called enhancers that allow for other proteins that will then complex with the transcription factors to allow the whole thing to happen even better. Remember, the DNA itself is not a rigid protein, or a rigid molecule. It's not a protein either. It's not a rigid molecule. It's floppy like a noodle, and it's like bending all over itself and folding along its length. So by folding then the enhancer region with its activator back over onto the promoter region where the RNA polymerase and the other transcription factors are, we can actually get that transcription to work even better. even though that enhancer region might be located quite a distance from the rest of the gene itself, it's still a way to control the gene based on what types of proteins are going to be able to interact with that enhancer region. There are activator proteins that are going to make the transcription go better, but there are also silencer proteins that will block the transcription if they complex with those transcription factors. Looking a little closer, we can get a little bit more complete of a picture of what it's going to take, the suite of proteins that it's going to take to actually turn a gene, a length of DNA, into messenger RNA. And it's more than just the RNA polymerase. And it's even more than all of these other proteins that have to complex together in order to make the transcription factors. But there are also other activators that are involved, and I don't have any silencers on there. This one is activated. But it gives you an idea of the complexity of the structure. So far, we've been discussing different ways that the environment can be affected to allow for DNA to be transcribed or not. We can also affect the DNA itself through methylation to make it also not really compatible with allowing those transcription factors to complex in the first place, effectively turning off the gene. We do this by adding a methyl group onto the cytosines, and this again turns off the gene pretty much permanently. This is how Barr bodies are produced through X chromosome inactivation. Of course, we know that once transcription has taken place, that messenger RNA is not done. It needs to mature, right? It needs to have all of those introns spliced out of it. And again, by affecting how the spliceosomes work and which sections they take out and which sections they leave in, I can have a gene that is expressed one way actually be post-transcriptionally processed to express a different way. And even if that messenger RNA is allowed to mature into the right form, it has to last long enough in order to make it to the ribosome to be translated. And remember, the cytoplasm is a dangerous, dangerous place. There's all kinds of nucleases out there that are getting ready to chew up any bits of nucleic acid that are floating around. And so not only when the messenger RNA is processed, not only do we get the coding region, but we also get that 5' cap in the poly A tail, right, to give those enzymes something to chew on before they get to the coding section. And of course, if I make a longer poly A tail, then that messenger RNA is going to last longer. It's going to complex with more ribosomes, and it's going to yield more proteins. Nucleases are not the only cytoplasmic hazard that messenger RNA has to contend with in order to become translated into a protein. Turns out there's other RNA out there that can also get in the way and keep that keep the messenger RNA from kind of doing its job. Small interfering RNAs are short sections of RNA that bind to the messenger RNA. They have a perfectly complementary base sequence, and that makes a double strand of RNA, which is weird, and there are these other proteins that will bind to the double-stranded RNA and essentially chop it up, keep that messenger RNA from surviving. And by doing that, you essentially turn off the gene because it's not making a protein. And even though it makes the messenger RNA, you don't make the protein and you get no trait. Now, there are other short sections of RNA that have some parts that are complementary to the messenger RNA out there, but it's not as good of a match. And so when the proteins come and attach to them, they don't break that. They don't break the RNA down. They just kind of like, I don't know, fold it up and store it up for later on. And so it might eventually degrade, but for now it's really repressed, and it can actually be released again and survive. So this does present another level of post-transcriptional control by not allowing that protein to be made just then. RNA interference has been certainly a hot topic in research, both for just understanding how genes are turned on and turned off, and as a potential for therapeutics. It won Doctors Fire and Mello here a Nobel Prize 2006. Dr. Takis at Yale is a college buddy of mine, and he has literally written the book on RNA interference in zebrafish in Danios. Even if that messenger RNA is able to survive the gauntlet of cytoplasmic nucleases and silencing RNAs and microRNAs, it still has to complex with a ribosome. And this presents another point at which gene expression can be controlled, can be turned off. By taking a big old honkin' protein and attaching it to the 5' end of that messenger RNA, I can essentially prevent the two subunits of the ribosome from complexing and allowing that transfer RNA to come in and start this whole process. And you end up with no protein. Once that polypeptide has been assembled, the amino acids are all in their proper order, it still needs to be processed. It still needs to be folded, and some parts cut off of it and other things added to it, and proteins added to it so it gets to the right place in order for that protein to kind of achieve its final goal of causing a particular phenotype. So this kind of post-translational processing gives me another place, or gives the cell another point at which it can turn off a gene or turn off the effect of a gene. There's also how long that protein lasts in the first place. I can take, well, the cell can take a protein, any protein really, and label it for breakdown by proteasomes by attaching a protein called ubiquitin to it. Ubiquitin then is kind of like the death tag for old proteins. It's a relatively short polypeptide, but it's enough. If it's there, then the proteasomes recognize the ubiquitin, and they kind of chomp up and break down the protein. These are the gentlemen, the researchers that are credited with having determined not just what it does, but also its structure. If the ubiquitin is the death tag, then the proteasome is the executioner. It acts kind of like a protein shredder. It just like chops up proteins into little short fragments. Then you can take those short fragments and use them to do other things with. To recap and review here at the end of the different control points along the assembly line of gene expression, right, we have the transcriptional levels, which is going to involve whether transcription can start or not, if is the DNA packed too tightly, are there transcription factors there, and do they have access to the DNA itself? Are they associated with enhancers or promoters? I don't know. Also, we have post-transcriptional control. This has to do with whether the messenger RNA has been processed and spliced, those exons and introns. How long is the 5' cap poly A tail? Is it going to last long enough once it gets into the cytoplasm? How much silencing RNA is there out there, or microRNAs, interfering RNAs are there out there? And it has to complex with the ribosome. Is there a 500-pound gorilla of a protein attached to the 5' end of the messenger RNA, preventing that complex of ribosome from taking place? And even after translation has taken place and we have the protein, or at least the polypeptide, that polypeptide has to be folded into the right shape to be a protein and have its effect. And that protein has to last long enough to actually do its job and have its effect. So if I ubiquitinize it, it's going to be degraded and still not have its effect. Oh my, that's enough information. If you have questions, and I know you have questions, please, please, please feel free to leave them in the comments section, drop me an email, or if you're doing this on Edpuzzle, you know what to do. Other than that, keep washing your hands. Hope you're all staying healthy, and I hope to see you soon.